and gentlemen, distinguished speakers and participants from around the world, good morning, good afternoon, and good evening. I am Jang Homin, Chief Director of the Korea Institute for Promoting Asia Biosafety Cooperation, KIPABiC in short. On behalf of KIPABiC and UNEP JEF multi-country project, Biosafety project, it's my great pleasure to welcome you to today's webinar on risk assessment and risk management, looking ahead to the 11th meeting of the parties to the Cartagena Protocol on Biosafety, CP-MOP 11. We are delighted to have you join us as we delve into the critical developments in the risk assessment management of living modified organisms, a key area of focus under the Cartagena Protocol on Biosafety since 2004. Today, we are expecting 700 participants from more than 50 countries representing government, academia, industry, NGOs, and civil society organizations. With the rapid advancements in biotechnology, particularly around engineered gene drives, today's discussions are more relevant than ever. Much progress has been made on gene drives recently, especially with the advent of CRISPR-based gene editing with wide range applications, particularly in insect species. At the upcoming CP-MOP 11, a voluntary guidance document on gene drives will be discussed. Additionally, new topics such as living modified fish will be addressed with the aim of developing further voluntary guidance to support case-by-case risk assessment of these organisms. This webinar aims to provide participants with the latest insights on these developments, engage experts in discussions on draft decisions, and prepare participants for the upcoming CP-MOP 11. Today's webinar is organized KIPABiC in collaboration with UNEP and 4 participating countries, Bangladesh, India, Mongolia, and the Philippines. This webinar is part of a capacity building and knowledge sharing initiative under the JEF country project titled Promoting the Safe Application of Biotechnology through Multi-Country Cooperation in the Implementation of National Biosafety Frameworks in Asia. This 3-year project with JEF funding of $1.09 million involves co-financing from 4 countries and the government of the Republic of Korea. UNEP provides technical supervision, while KIPABiC contributes as the lead executing agency and project management unit. To share a bit more about KIPABiC, we are a non-profit organization based in South Korea, dedicated to supporting countries, primarily in Asia, through capacity building initiatives and collaborative approaches in biosafety and biotechnology to support parties to fulfill their obligations under the Cartagena Protocol on Biosafety. For today's webinar, we have 1 technical session consisting of 5 technical presentations, followed by a Q&A session. We are honored to have renowned experts in this field with us today to share their expertise and provide up-to-date insights on the latest developments. We sincerely thank you for taking the time to join us and for your support of this initiative. Before I hand over to our moderator for the session. It's my pleasure to introduce Dr. Alex Owusu-Biney. While Dr. Alex needs no introduction, allow me to briefly share the following. Dr. Alex serves as the UNEP JEF Portfolio Manager for Biosafety, with over 2 decades of experience in biotechnology, biosafety, and policy advocacy. He oversees the development and implementation of national biosafety frameworks globally, and brings extensive expertise in molecular biology, regulatory science, and project management. Thank you for accepting our request to be our moderator. Dr. Alex, I invite you to take the lead in moderating this session. Thank you, Dr. Alex.
Thank you very much, Dr. Homin Jang. And I want to use this opportunity to welcome all the presenters again and the participants. As most of you are aware, under the Biosafety Protocol, risk assessment and risk management are 2 critical thematic issues that forms the anchor and the platform for decision-making. It's therefore important that our national or regional systems have very well developed risk assessment, guidance, or technical regulations, which can guide and provide needed scientific support to decision makers based on the science at the particular application or moment. With that, I want to just give a quick overview of how this session would go. We will have the 5 presentations back to back before we take questions and answers. The organizers have created a patch where there's a Q&A place where you can put your questions so that you don't forget before any of the presentations end. The presenters will, Dr. Vibha Ahuja will give a 10-minute overview. I'll come back to it, and then the other presenters have 15 minutes each to make their presentations. If they save time, good for all of us, but we'll try to work within the time to give enough time for discussions. Our first presenter today is Dr. Vibha Ahuja, who is an expert on biosafety and regulatory aspects and has been around in this area for more than 25 years. She is well versed in the Indian biosafety regulatory system and also has been involved in evolving from guidelines on including risk assessment, not only in India, but in the South Asia region. She led preparation of biosafety documents on dossiers for decision-making even in the Indian system, and has also of late been involved in policy issues related to gene editing in India through her engagement with the Indian National Academy of Agricultural Sciences, and is a member of the drafting committee constituted by the Department of Biotechnology. I think for a lot of people, Dr. Ahuja is not a stranger. I'll pause here and invite Dr. Ahuja to give the first presentation. I'll come back to each speaker as they present. Over to you, Vibha.
Thank you so much, Alex, for introducing me, and thank you to KIPABiC for inviting me to provide this overview in the beginning to set the stage for the next presentations. So I will quickly walk you through what has been the developments under the risk assessment, under the protocol, and then we will hear from the other speakers on the current issues. So I just want to check my slide is available in the, you can see it in the slideshow mode, hopefully. It's okay?
Yes, it's okay.
Okay. So because we have a very wide audience and some of them are not familiar with the Cartagena Protocol, just like to briefly introduce that this is a protocol which was negotiated under the Convention on Biological Diversity, adopted on January 29, 2000, and it has 173 countries as the parties. As on date, 10 meetings of the COP-MOP to CPP have been held, and now the meetings are held concurrently with the COP to the CBD. The objective of the protocol is to ensure an adequate level of protection in the field of safe transfer, handling, and use of living modified organisms resulting from modern biotechnology that may have adverse effects on the conservation and sustainable use of biological diversity, taking also into account risks to human health and focusing in particular to the transboundary movements. So these are the various, key elements, what we call of the Cartagena Protocol. The first is the decision-making procedures. As Alex already said in the introduction, risk assessment and risk management is one of the critical elements based on which the decisions are made, and in the protocol all the things move for the use of biotechnology. Then handling, transport, packaging, identification, and information. But RARM is one of the very important topics in the whole thing. And this is covered under 2 articles within the Cartagena Protocol, Article 15 and Article 16, which we call. Article 15 is the one which establishes the basic requirements for risk assessment, and it refers to what we call the Annex 3 for further guidance. Annex 16 deals with the management of risks of the organisms that fall within the scope of the protocol. The most important with respect to the operationalizing of the risk assessment frameworks in the country is Annex 3, which sets forth the objectives of the risk assessment, what the risk assessment, what are the risks, general principles that the risk must follow, methodology, and in particular points to consider for assessing the potential impact of this, risks of this. So if we look at the key components on how Annex 3 guides us, the risk assessment methodology, then it can be translated into 6, key components. The first one being the hazard identification, then exposure, likelihood assessment, hazard and consequences of that hazards, and risk characterization. What are the risks that we finally, identify, and then the management of that. So this whole approach is very nicely provided for in the Annex 3 and has been consistently used by the most countries where these products have been approved and are under use for several countries. And this particular approach defined in the Annex 3 has been, as I said, successfully used for a wide range of organisms. So how do we further operationalize it? Very quickly, it is based on the comparative approach, and we normally call it in the paradigm of risk assessment, it is referred to as problem formulation. So we do this comparative assessment and molecular characterization, try to identify what is the difference between the GMO and comparators, and this is what is the problem formulation. Important considerations here are the familiarity with the organisms, and then finally, are these differences likely to have a significant adverse effect on the valued entity? So this is what we call the problem formulation, which is the very, very much an accepted concept now, and has also been adequately tested. So just walking you through what has been the development since 2003, when the protocol came into force. To start with itself, there was an AHTEG which was set up to understand the scope of nature and scope of existing approaches and identify any gaps so that the parties can implement these articles efficiently. I'm not going to go through this step by step, but just want to share with you that from 2004 to 2016, there has been a very elaborate process for the development of what was called a guidance on the risk assessment of LMOs. There has been a lot of divergent views among the parties, which led to a long process where this document was drafted, expert groups were set up, the testing was done, and then there was also a new AHTEG was set up which analyzed the testing. And finally, what the parties agreed was what is called a voluntary guidance, voluntary document, for consideration by the parties, which they can use as per their circumstances. So this is very important to note that, that's how you can correlate why we are saying voluntary documents for gene drives. So then there has been, what you call the request from the parties, and also as part of the process, parties have been requesting for preparation of additional guidance documents from time to time, and this list keeps growing over the years. However, as I said, risk assessment and management has not been a very easy topic, because the considerations there are divergent views, and there were divergent views among parties on whether or not additional guidance on specific topics is required, or that basic principle, basic document, which is based on problem formulation, is applicable. So based on this, discussions, a process has been established under the protocol. It was done in CP-MOP 11 for the identification and prioritization of the specific topics regarding the risk assessment of living modified organisms, and also consider whether, every topic which comes, this process has to be followed to consider whether this additional guidance material are needed or not, to put rest to the discussion. 1 minute, Mike. Okay. So what are the criteria in this process? It's very important to know. These criteria are that first of all, any of those topics should have been identified by parties as priorities, taking into account the challenges they're facing in the risk assessment. It should fall within the scope and objectives of the CPB. And most importantly, as I said, problem formulation approach has been followed, and several LMOs have been approved. Of course, my colleagues later will be talking more about it, but we have to see whether there is a challenge to the existing risk assessment framework, guidance and methodologies, and then we have to see if there is any specific technical or methodological challenge, then there will be a need to come up with a new guidance. Otherwise, the main document follows. So subsequent to this 2016 document, as I said, the attention was on this process for identification and prioritization was set up. And then there was an AHTEG set up to analyze the need for additional guidance on two topics, that is the LMOs containing engineered gene drives, which were thought to be having a slightly or maybe a different approach, or maybe additional considerations to the existing approach, and LM fish. So subsequently, in the next COP in 2020, it has been decided to set up an AHTEG for guidance on engineered gene drives, and not to proceed with the guidance for LM fish at that time, and it was to be considered at COP 11. Parties were also asked to submit additional topics. So based on all these, discussions, I just wanted to give a glimpse of what has been there in the risk assessment, area under the protocol. The issues of discussion which are there at COP 16 MOP 11 is to consider the additional voluntary guidance materials to support the case-by-case risk assessment of LMOs containing engineered gene drives. This is one of the guidance material which has been developed by AHTEG, and has to be, the parties have to consider this. Of course, there's a need to conduct capacity building activities in support of risk assessment, and there is a draft decision, I hope it will be a draft recommendation from SubSTA. Then to establish an AHTEG tasks with development of additional voluntary guidance materials to support the case-by-case risk assessment of LM fish. This is another topic of discussion on various parties have different opinion. This had already gone through some process and probably has to go again. And then to identify the list of prioritized topics for preparation of additional guidance material. So this is what is from my side, and brief overview of the discussions under RARM under the COP so far. Thank you so much.
Thank you very much, Dr. Vibha Ahuja. As I said, I will attempt not to comment on the issues. I will just moderate the discussions as much as possible. So thank you once again. But it presupposes that those who are going to go to the COP must really prepare, look at SubSTA recommendation 26.5 and a draft decision, and look at the voluntary guidance also, so that you are in a position to make informed decisions during the negotiations. Thank you. Our next presenter is Dr. Flerida Carino. She's going to talk about environmental risk assessment under the Cartagena Protocol on biosafety. Dr. Carino is known for her extensive work with insecticide-resistant genetics, biochemistry, and molecular biology. Dr. Carino has been involved in drafting policy and regulations for GMOs since 1994. She is currently a consultant to the Philippines Food and Drug Administration, providing guidance on urban pesticides and risk assessment of GMOs and their products. She served as faculty in several local and international training programs on food safety and environmental risk assessment for several international organizations involved in biosafety capacity building and for various Filipino government institutions involved in the regulation of GMOs. Dr. Flerida Carino, you are welcome to make a presentation on environmental risk assessment. Over to you.
Thank you, Alex. We meet again. We've been meeting each other in these webinars as well as in many negotiations. Let me share my screen. And let me play with this. Let's see. Hopefully I'll be able to. Why is it not working? There. Can you see my slides?
Yes, please.
Okay, thank you. I'd like to thank Vibha for the introductions that she's given on the environmental risk assessment in relation to the Cartagena Protocol on biosafety. And this time I'd like to discuss an approach for environmental risk assessment. That's actually something that we normally would do for pesticide evaluations, for the safety of pesticides, especially for urban environments. So I'd like to discuss something about the problem formulation approach for environmental risk assessment. And you'd notice that I did not say that this applies only to LMOs, because I think it would apply to many other situations. The Cartagena Protocol on biosafety has already been discussed by Vibha, so we won't go into that in greater detail. Let me just say that this discussion will be in relation to the Cartagena Protocol on biosafety, abbreviated as CPB, in contrast to the Biodiversity Protocol, which is CBD. So the alphabet can be quite confusing, but the CPB is our major topic for today, and the approach is the problem formulation approach. One of the features of the CPB is to protect biological diversity from the potential risks. It aims to ensure the safe handling, transport of living modified organisms. Its objective is to evaluate and identify the potential adverse effects of LMOs on the conservation and sustainable use of biological diversity. And if you look at this particular objective, you'd notice that what we have as the protection goal is the conservation and sustainable use of biological diversity. And it begs the question that since the environment is variable and the LMO can also be variable, assessment should be done on a case-by-case basis. Annex 3 gives you a brief methodology for the deliberate release of LMOs into the environment. It requires the four tasks to be made: identify the novel genotype and phenotypic characteristics of LMO, the evaluation of the likelihood of adverse effects occurring, evaluation of consequences if adverse effect is realized, and estimation of overall risk considering likelihood and consequence. For a long time, we had not had any international document that recommends an approach for doing an environmental risk assessment for LMOs. But the recent issuance of the OECD in 2023 essentially recommends the problem formulation approach in looking at possible considerations when you want to release an LMO into a given environment. I'd like to do my acknowledgements before I proceed, because I have borrowed heavily from other presentations made by Dr. Monica Garcia Alonso, Don McKenzie, and Dr. Andrew Roberts of the AFC from the biosafety training workshops we have done in several countries already, and we have used some of these slides before. So I'd like to acknowledge your contributions to this presentation. Problems formulation approach can be summarized in the following figure. So you have to identify your protection goal, gather relevant information, do an initial risk characterization, and either come to a conclusion or a risk characterization that will tell you risk is very low or no risk at all, or you come up with the identification of missing information. So in a nutshell, this is actually how the problem formulation approach can be utilized to augment our appreciation of environmental risk assessment that we could do for LMOs. If you remember, the protection goals that are defined by the country regulations as part of national policy, or in our particular case, the CPB, are often formulated in legal terms so that we have normative concepts as sustainability, integrity, acceptability. This can be very, very difficult to interpret and therefore often impossible to prove or disprove. As a result, it can be too vague for scientific assessment, considering that a risk assessment is a focused process that will utilize a scientific approach so that you can come up with a specific hypothesis and you can support that by appropriate testing as well as other available information. Pardon me. Now recall some of the features of the CPB. It says protect biological diversity. So protect biological diversity can be interpreted in so many ways. And so if the objective is conservation, sustainable use, again, these are normative terms, which are very hard to delimit. And as a result, we'd have to use a problem formulation approach to brief, sorry. I'm sorry about that. I'm suffering from the flu. So these are very normative terms which are difficult to define as well as delimit. And as a result, you'd notice that if you look at Annex 3 of the Cartagena Protocol on Biosafety, that gives you the hazard identification, exposure assessment, hazard characterization down below, there's always a difficulty in looking at hazard identification as being the most relevant aspect that starts this downward triangle. So hazard identification is one of the hardest things to do because you can identify 1,001 possible hazard, but you'll have to limit it to one that particularly applies to a given situation, a given LMO, given host, given donor, as well as the given receiving environment. The protection goals are usually very broad and not always clear. As a result, you'd like to distill this high-level protection goal into an operational protection goal, which can allow you to form a testable hypothesis that will give you at least an idea of how to do your risk characterization. So let's look at this. If you look at protection goals in the CPB, biodiversity is one of its goals. So we'll just look at the top of this figure. Okay. Biodiversity is one of the goals of your CPB. The policy protection goal is protection of biodiversity. That is what the country policy is. That's also what the CPB policy is. Operational protection goal is the aspect in the policy that will be included in the risk assessment. And once you've identified the operational protection goals, which can be multiples, you can end up with an assessment endpoints that will define what will be assessed based on the characteristics of the product and the potential harm that it can cause. From then, you can determine the measurement endpoints to be able to ensure that you will be measuring actual relevant details to assess risk for chosen assessment endpoints, and this is based on a testable hypothesis. So you can see from this figure that you go from the very broad to the very specific. Okay, so let's have a few examples to look at how it's going to be done. Operational protection goals will allow you to provide more details on what aspect of the environment are to be protected from what, where, and in which context. For example, protection of biodiversity. So let's have an example of a GM that contains, let's say, an insecticidal protein. So you can say that the operational protection goal should be that the cultivation of the GM crop X should not result in adverse effects on non-target organisms of key conservation value in off-field areas compared with cultivation of non-GM varieties of this crop. So one of these goals, protection of biodiversity, can now be distilled into an operational protection goal. As I said, you can have multiple operational protection goals. You have further distill the operational protection goal into an assessment endpoint. So if the operational protection goal is given, the population decline of valued and non-target organisms in off-field area during cultivation of GM crop of the following season can be your assessment endpoints. So you've defined an assessment endpoint to provide detail on the entities to protect. In here, you have to specify what is your non-target organism, what is your environment, what is the temporal and spatial scales where exposure of your organism can actually be occurring. And then from your shall we say assessment endpoint, you can formulate a test hypothesis such that exposure to insecticidal protein A produced by an LMO will have a greater adverse effect, say, on the population size of a protected species of NTO, for example, the monarch butterfly, than the cultivation of non-GM varieties of the crop, in which case you have now identified your measurement endpoints, the potential exposure, that is temporal and spatial overlap, and levels of exposure of your non-target organism to toxic protein A. Sorry. Okay. Using problem formulation for ERA gives you the following. Once you have identified your protection goal, okay, it will identify the information that you need for assessment. And when you start gathering the relevant information, you'll start categorizing what information you have, identifying whether, shall we say, determining whether or not you have enough information, and determine what additional information you still have to have. So you can either reach a conclusion or identify the missing information so that you can input into the initial risk characterization and do this iterative process all over again. Now, gathering relevant information can come from various sources. It can come from the information gathered during the development of the product itself, the regulatory package prepared by the developer, risk assessments conducted by other regulatory agencies, information gathered during commercialization of the product in other countries, information gathered by public research institutions and published in peer-reviewed papers. Any information can be considered, provided they are of high enough quality and they are verifiable. So, existing information, the host organism will give you familiarity. Existing information, donor organism will also tell you what the genes are supposed to be doing. The information on the LMO gives you the additional trait that your LMO would have. And existing information, receiving environments will tell you about the interaction of your LMO with your specific environment. Now remember that risk characterization, that risk is, when you're doing your risk characterization, risk is a function of hazard times exposure. If there's no exposure and no hazard, then you don't have to worry because the risk is quite low and therefore quite acceptable. If the exposure levels are not known or the level of hazard is not known, then you would require more data to come up with the risk conclusion, as is shown in the inset over here, that tells you that this is indeed an iterative process. You have to keep going back so that you can come up with a good risk characterization that will allow you to be able to use this risk characterization matrix. If the hazard is going to be marginal up to major, and exposure is going to be highly unlikely to likely, can add up with a matrix that will characterize your risk as being negligible or major. So you can use this table so that based on your risk characterization, informed decisions can now be made. Now, taking into consideration what has been required by the Cartagena Protocol, the problem formulation approach will allow you to focus your risk assessment on what is truly relevant for your major concerns as defined by your national policy, and it's in this particular case by the Cartagena Protocol on biosafety. So what you've done when you use a problem formulation approach is limit your risk assessment, focus your risk assessment to what is really quite relevant to the problem at hand, and actually help you do your hazard characterization in a manner that is scientifically as well as scientifically sound and highly defensible. So thank you very much, and I'd be willing to answer the questions as we go along. Thank you.
Thank you very much, Dr. Carino, for your presentation, setting additional technical inputs from Vibha's presentation. We all know that you cannot do a risk assessment training in just 15 minutes. So we are just giving you pointers. Those who are interested will need to seek further training in this area. She also made a very important point. Risk assessment is not unique to a biosafety protocol. Actually, all the principles we use are quite similar in pest risk analysis, even in food safety assessments. The difference is that you do it in the context of the object of what you are looking at for the decision making. So in that context, we have a lot of expertise in countries, but sometimes government systems push out academia and other aspects. It's important that as you even go for these negotiations, you involve your national expert teams from government, from private sector, to have a clear picture and position on some of these issues. Otherwise, it becomes the normal talk shop. It does not bring out anything but more complaints after COPs. On that note, thank you very much. Once again, we will go to the next presentation as we delve deeper into this webinar. The next presentation will be by Dr. Brinda Das, and she'll be talking about the guidance on risk assessment of LMOs with engineered gene drives. Dr. Das is the gene drive policy lead at Foundation for National Institutes of Health, the US, and a member of the AHTEG who attended the SAPTA 26 in Nairobi. Dr. Das has a PhD in cell and molecular biology, a master's in public health, and extensive experience in transgenic laboratory animal care and use, preclinical drug testing, clinical trial management, and peer review of new animal drug dossiers for genetically engineered products. The other areas of interest includes regulation of and policy development for engineered insects intended for population suppression, disease mitigation, invasive species, stroke environmental control. Dr. Das, somebody was already asking a question about engineered insects, gene engineered. So this is a very opportune time as you delve into this presentation. Welcome, and over to you. Thank you.
Thank you so much. Thank you to organizers, and thank you also, Dr. Alex. Let me share my screen. So hopefully all can see my screen, and in the interest of time. So I just want to give a quick overview of what gene drives are. Gene drives themselves are a naturally occurring process. They occur typically in sexually reproducing organisms, and it leads to a particular genetic sequence in the genome that is inherited in a biased fashion. That means it's inherited more often than you would expect by Mendelian frequency. So the interesting thing is, even if there is a fitness cost associated with the sequence, it will still be inherited in a biased frequency. And what this means is that the sequence essentially is present more widely in the population more quickly, so within a few generations. And of interest to us with this COP in mind is that right now the products that are most ahead in development belong to insects, so genetically modified mosquitoes that contain engineered gene drives. So there are two main types of applications being looked at right now for these types of products. One of them is population suppression, where as you release the insects and they spread the trait, and the population that is in the environment will decrease in number, and eventually the goal is for it to disappear. These are typically for pest species, whether they are vector-borne disease transmitting organisms or pests of crops. And then the other main type of application is called population modification. And this is where the population number doesn't change in the environment, but the population is replaced eventually by the gene drive, engineered gene drive containing population that is released. So in terms of gene drives, as I said, they are naturally occurring phenomenon and sequences, and the goal is to harness that methodology that exists in nature and engineer organisms to have a trait of interest be biased in when the GMOs release. So there are interests beyond pest species of insects. There are interests in rodent control, invasive species control. There are also interests in human health and agriculture. So the example that I will give is, as I said, the most advanced right now, but that's not to say that there are not other groups interested in other types of engineered gene drive organisms. So, but going to a COP-related topic, I just want to give a quick background, go back to COP Cartagena Protocol MOP 9, which was in Egypt in Sharm el-Sheikh. And that COP identified 2 topics for further guidance on risk assessment. One was for living modified fish, LM fish. The other one was for engineered gene drive organisms. And as is quite common in this process, there was online forum. And unfortunately, because of the pandemic, the AHTEG was held as a virtual format for the first time. In 2020, there were 2, if you look at the decision 913, you will see that there were 2 documents that were prepared. One was on LM fish, the other one was on the gene drive organisms for consideration by the online forum and the AHTEG. In the end, after the AHTEG met, the recommendations of the AHTEG was that there was no unified recommendation for need for additional risk assessment guidance for LM fish. On the topic of engineered gene drive organisms, it was agreed that these synthetic or engineered gene drive organisms do fall within the purview of Cartagena Protocol, and there were concerns raised about whether there would be adequate community engagement and IPLC perspective, since a lot of these fast developing products are aimed at use cases in developing economies. There was, of course, a lack of data because these products have never been developed before, so they have never been tested, they have never been trialed in field or, of course, put on the market. And this remains so even today. So there's no history, what we call history of safe use. There's no history at all. And there's some capacity issues with scientific and technical capacity because this is a new topic as such for biotechnology. And then there were other allied questions about the ability to monitoring methodology, modeling, and the space for modeling in this area, as well as the spatial scale of spread and dispersal. Overall, the recommendation coming out of this AHTEG to be considered by the next MOP was that there was a need for additional guidelines for risk assessment of engineered gene drive organisms. So going to the next COP, which would be MOP 10 for Cartagena Protocol, this is 2022. It was agreed coming out of this meeting that we would have a AHTEG to look at proceeding with drafting an additional voluntary guidelines for living. It was agreed for engineered gene drive organisms, but not for LM fish. So LM fish topic was dropped for that time. And so again, there was an online forum. There were 2 of these. There was an ad hoc technical expert group formed, as is conventionally done under these circumstances. And we had 2 AHTEG meetings in 2023. 2 forums convened in 2023. And then subsequently, we had 2 AHTEG meetings as well, one in 2023 in November and another one in January. And there was a outline provided by ICGB, and this was discussed in the first meeting. And then there were working groups for drafting sections of the document. Because of the expedited timeline, the drafting groups worked and created the documents. The documents were put together by the Secretariat and was recirculated as a whole document back to the whole AHTEG. and comments were collected, and the AHTEG met to discuss in plenary the document, the final document, which was finalized and provided for Substa 26. So the document itself, it's available for your consideration ahead of COP. As Dr. Alex mentioned, please do go and read the document so that you come prepared. It has primarily 7 sections in terms of risk assessment itself is in section 4, and you will see that a lot of the issues that were raised in the previous CP-MOP and previous AHTEGs about need for additional information on benefits, IPLC, free prior informed consent, public participation. These are also covered in the document, even though they are not strictly methodology of risk assessment, but they are covered because they were raised as issues. The document itself has also annexes which go over some of the topics that are specific to a product that has no history of use and is very new to being used globally, worldwide, right? And then it also goes over, it gives examples of current engineered gene drive insects, because the focus is particularly on mosquitoes, as was required by the decision document 1010. For those of you who are interested in what this means for COP16, just to reassure you that this additional voluntary guidance is science-based, it still takes the values of Annex 3 forward. It's based on Annex 3, so it's science-based. Asks for the risk assessment to be on a case-by-case basis, using, as Vibha described, the comparative approach. It's transparent and iterative. Again, features that are, for all risk assessments, regardless of the motivation to do one. And it's more specific to engineered gene drive organisms and is in line with WHO guidelines for phase testing. It also leans heavily on OECD documents when it talks about familiarity, tiered testing. And as Dr. Flerida mentioned, it's based squarely on problem formulation. So I won't go too much into problem formulation because it's already covered, but I just wanted to show you on the right of your screen is a figure that I took from the additional voluntary guidance that, as I mentioned, it leans heavily on Annex 3 of Cartagena Protocol. So it's very much aligned with Annex 3. You can see it has taken problem formulation and overlaid it on the steps of risk assessment. And these numbers on the side that I highlight 8A, 8B, these are the subsections, the paragraphs from Annex 3. And you will see that they align perfectly with what Vibha described as a risk assessment process: a hazard identification, exposure assessment, hazard characterization, risk characterization, and finally, the risk report that will be provided to decision makers to make a decision in country. So to tell you that it's taking existing standard of practice from risk assessment field, especially what is used for GM crops, and aligning well with Annex 3 to also show you how it can be extended to use for engineered gene drive containing organisms. I don't want to go too much into problem formulation because Dr. Carino did such a good job, but I just want to tell you that this document as well, it shows you how to use problem formulation to scope and define the boundaries of the environmental risk assessment for engineered gene drive organisms. It talks of many of the same things that Vibha has described: comparators, assessment endpoints, protection goals, how to bring the stakeholder concerns into the protection goals. So how to operationalize not just your law and legal aspects, as well as your international treaty obligations, but also how to use community concerns and bring them into the protection goals. And ultimately, the goal is to have a decision support tool that countries can use to make a decision that supports their country needs. The document, this is again taken from the guidelines themselves. It goes into examples. It shows you both how to go from the area of protection to define the protection goals to what you would actually consider an assessment endpoint, and then more critically, how to translate that into something that is quantifiable or measurable so that you can track the results of your work, right? And this is also taken from that same document. This shows you an example, just to give you an idea of how you can create a pathway to harm. This is a pathway to harm that was created by the AHTEG in plenary. And you can see this is one particular pathway. And there are other ways that you can look at using similar pathways, adverse effects to human or animal health, and adverse effects to the environment, which would feed into protection of the ecosystem and biodiversity. As I mentioned, the document is specific to engineered gene-driven mosquitoes because this is what the terms of reference was for the AHTEG. So it goes into mosquito-specific issues, which are inserted in boxes in the text of the document as required, as relevant. And you'll see that it shows you examples of different types of gene drives. So you have both the spatial scale as well as temporal scale, and then self-limiting and self-sustaining. And it also shows you currently what types of gene drive mechanisms are being developed in the lab. And I will reiterate, these are all still in highly contained environments. There is no gene drive being released anywhere in the world. So in terms of risk analysis for engineered gene drive containing mosquitoes, the main thing that people are concerned about, because mosquitoes can fly, is spread and dispersal. So spread is when it spreads within a population, within a contained population, and dispersal is when it moves from one population to another. So it disperses from one area to another. But I want to reassure you that this document shows that there are common themes and that the risk assessment paradigm still applies, regardless that it is a mosquito or that it is engineered gene drive containing mosquito. Essentially, you still want to be case by case. You want to take into account your stakeholder concerns. You would like to work ideally with your stakeholders in the areas where you want to do the releases or the work, and co-develop the material with them. You want to do what Vibha suggested, operationalize the protection goals, use the problem formulation methodology, which it describes, and then you would want to do the pathways to harm and use tools that are current, like modeling and a phase testing approach. And then, of course, it also describes monitoring and risk management that would be involved at the end of the pathway if you were to release it in the field. So in terms of recommendations, going back to decision document 1010 from 2022, it would be good if the outcomes were welcomed and the document was endorsed, and parties could use these documents and train in capacity building to make this document actually a living document which is being used and is useful in the field. And as I mentioned, this would be really helpful for both the regulators and decision makers as well as developers, so there's a clear idea of what is required in terms of data and what would support a decision that was informed and science-based and risk-based, so that the countries that need these types of products can actually be the judge of whether they feel it is safe and effective enough for them to take on board. As well, there was a request to establish an AHTEG for the risk assessment of LM fish. I just want to note that this topic has already been discussed by a prior AHTEG under 913. And these are the tasks that are mentioned in the recommendation for you to look at for COP, is that the AHTEG should meet, and then they should consider whether there are other topics that need to be prioritized for risk assessment, including LM fish. And of course, there will be an online forum, and the Secretariat will be synthesizing the materials. As I mentioned, LM fish is a topic that has been settled by a prior AHTEG, and the decision there was that there was no need for additional guidance. This was a decision that COP agreed with as well. So far, in my opinion, I don't see any new reasons to reprioritize this topic, but of course, COP will make that decision for the future AHTEG. And this is something also that Vibha mentioned, that there will be, of course, parties will be submitting a list of needs and topics for further guidance. So please, parties, you should consider what it is that you need capacity for and approach COP with that in mind. Of course, your scientists and your focal points will be the best people to lead you in that direction. And then we will follow through, of course, with the Secretariat, prioritizing those for online forums and AHTEGs. One point I want to highlight is that the synthetic biology multidisciplinary AHTEG has invited parties to consider self-limiting insects as a potential topic for risk assessment guidance. But I don't see a definition of what is included under self-limiting insects. And there are already GMO self-limiting insects on the ground. There are several countries that are using them both commercially as well as on a trial experimental basis. Recapping what has already been agreed by AHTEG and COP, there's no requirement as such for LM fish risk assessment guidance or any other LM insects, because they can use the one that has been created, the draft additional voluntary document that has been created by the current AHTEG. And that problem formulation approach is sufficient for all LMOs on the market, regardless of species. Of course, there will be some case-by-case requirements for additional types of data and additional types of methodology, but that doesn't mean that the problem formulation approach is not valid. So I leave this here. I don't want to go over this, but this is taken from Decision 913, maybe not exactly verbatim, but the concept is the same. This is how the topics, for prioritization were considered as per Decision 913. So I end here. Thank you.
Thank you very much, Brinda. And colleagues, she's gone through some of the issues on gene drive-related risk assessment, and she's also pointed us to further work to prepare ourselves for the COP. I believe, based on your questions, we will respond. And as much as possible, a lot of these colleagues are still going to be available. So if you have follow-up questions when the materials are shared, you would be able to get access. There's an important point that Brinda made. Sometimes some of the answers we are looking for are within country, but because a person is working with the university or a private sector company or some other research fellow, we tend not to ask them questions, but rather want to ask outside. The answers may be next door also. So let's use all the available resources to understand, in a bid to meet our obligations, whatever these experts are doing, whatever colleagues are doing, the obligation is the country which is a party, or in some cases, some can even non-parties see them as obligations. So you have a lot of resource, and we are always willing to be as possible. Thank you very much, Dr. Das. We will turn to a colleague from the CBD. As you all know, the CBD is the Secretariat for the Biosafety Protocol. And per Article 29, the CBD serves the parties functionally, but is administratively hosted by UNEP. So the CBD has two roles: administrative role of support from UNEP as they host the Secretariat, but they are functionally responsible to you, the parties, and your bureaus. So you provide, it's not a CBD telling you what to do, you provide what you want to be done. And that's extremely critical. Sometimes we get questions about, why are you not doing this? Secretariat can only work on what you request or guide. On that note, I would introduce our colleague who's going to make a presentation on our preparation towards CP-MOP 11, some of the key issues related to risk assessment and risk management. Because it's my colleague from the same organization, I'll say Austein without mentioning his second name. He's a program officer at the Secretariat. Austein works on risk assessment and risk management of living modified organisms under the protocol, as well as synthetic biology under the convention. In his previous role at the Secretariat, he worked on the His educational background is in molecular biology and genetics from University of Manitoba in Winnipeg, Canada. There he worked on RNA interference technology and mitochondrial antibiotic efflux. Austein, over to you. Thank you.
Thank you so much. I would like to first start by thanking the organizers of this webinar for inviting the Secretariat. We really appreciate the opportunity to support parties in this regard. I also wanted to thank the moderation of this webinar as well as the previous presentations that came before me. I will now share my screen. Perfect. As it was mentioned, I will be providing a brief overview of the preparations for the 11th meeting of the Conference of the Parties, serving as a meeting of the parties to the Cartagena Protocol on Biosafety. And I will overview what's been done during this intersessional period, as well as provide a brief background. So just as a background, in decision CP-913, the CP-MOP requested the commissioning of 2 studies, one on LMOs containing engineered gene drives and one on living modified fish to inform the process that was also established in that decision, in Annex 1 to that decision, for the identification and prioritization of specific issues of risk assessment of living modified organisms that may warrant further consideration. An ad hoc technical expert group was established, and they were tasked with reviewing the studies, performing an analysis on both LMOs containing engineered gene drives as well as LM fish. And the studies were requested to gather information on this process that is in Annex 1. So as previous speakers have mentioned, and as I'm showing on the screen, this is the process that has specific criteria, including that they're identified by parties as priorities, taking into account the challenges of risk assessment. They fall within the scope of the Cartagena Protocol. They pose challenges to existing risk assessment frameworks, guidance, and methodologies. And the challenges are clearly described, and also considering specific issues of living modified organisms that may have the potential to cause adverse effects on biodiversity, may be introduced into the environment deliberately or accidentally, have the potential to disseminate across national borders, and are or likely to be commercialized or in use somewhere in the world. And also consider a stock-taking exercise to determine if resources on similar issues have been developed by national, regional, or international bodies. So the 2019-2022 intersessional period started with the submissions of information process, as well as these 2 studies. So the first one was commissioned and was awarded to the Perseus, so Smits and Riddlesham, and then one on LM Fish, so JT Environmental Consultants by Mr. Sweet. So drafts of these studies were made available for the online forum, which served as a review mechanism before the studies were finalized and provided to the ad hoc technical expert group. Due to the timing, and as Dr. Das has indicated, it was a virtual ad hoc technical expert group, one of the first in the Secretariat. But the AHTEG did work well, and they were able to review the studies and performed an analysis on both of these topics, both the engineered gene drives and the LM fish. Regarding the engineered gene drives, there was a recommendation to develop guidance, but regarding the LM fish, there were a range of perspectives that were noted. And then working towards the last conference of the parties, serving as a meeting of the parties to the protocol in Montreal. In decision CP-1010, the CP-MOP endorsed the decision to develop additional voluntary guidance materials to support case-by-case risk assessment of LMOs containing engineered gene drives. An ad hoc technical expert group was established to develop these materials. Parties were invited to submit information on their needs and priorities. So this is that process in Annex 1 to decision CP-913. And then to consider the development of further guidance on the topic of living modified fish at the upcoming 11th meeting. The executive secretary was requested to provide dedicated web pages on both LMOs containing engineered gene drives and LM fish, as well as to commission a detailed outline to support the work of the AHTEG. So in terms of this past intersessional period, this is an overview of essentially the activities that have been done. So it started with a submissions of information. So this submissions of information sort of had two elements. The first was to collect information, to have parties, organizations, IPLCs, other governments support to submit information on engineered gene drives to support the work of the AHTEG. And then the second element is that the needs and priorities, so that process in that annex of decision CP-913. And then the first round of the open-ended online forum was convened to also provide information on engineered gene drives to support the work of the AHTEG, as well as to inform the drafting of the detailed outline. The Secretariat commissioned the International Center of Genetic Engineering and Biotechnology to draft this detailed outline. So the second round of the open-ended online forum was convened to review the outline, which was then further worked on by the International Center of Genetic Engineering and Biotechnology to finalize that detailed outline to provide for the first meeting of the AHTEG. At the first meeting of the AHTEG, the global experts took the detailed outline and decided to restructure it and to agreed to follow our problem formulation approach. And once that was decided, regionally balanced drafting groups were then established to work on various sections of the additional voluntary guidance materials. And given that time was short, the drafting groups unfortunately had to work over the holiday season last year, but I just wanted to also highlight the Secretariat's appreciation for all the experts because they worked hard. They had their own internal peer review process to provide comments on the final text, working towards that second meeting of the AHTEG. Also during this period, there was a second call for submissions of information regarding the needs and priorities for parties to submit on risk assessment. And then these two fed into the final meeting of the ad hoc technical expert group. In this, the experts finalized the text of the additional voluntary guidance materials, and then also considered the needs and priorities that were submitted by parties. The process then fed into SEPSA 26, and then given the time between the last meeting of the AHTEG and the 26th meeting of the subsidiary body, there wasn't time for formal editing of the document. However, the materials were translated and made available in the 6 official languages of the UN. And then following SABSTA, the document was formally edited and has been made available for the 11th meeting of the Conference of the Parties, serving as a meeting of the parties to the protocol. So just to highlight, and as other speakers have mentioned, the additional voluntary guidance materials has 7 sections and several annexes. So the first is a general introduction, sort of the objective and the scope, and then this general introduction, and then a section specific to engineered gene drives, followed by the general risk assessment considerations for engineered gene drives, followed by a recommendation on the acceptability of risk and the identification of risk management strategies, followed by monitoring, and then related issues. And as Dr. Das has rightly pointed out, these are issues related to risk assessment, but are key issues and part of the overall decision-making process. Then there are several annexes that include modeling, uncertainty, information on the WHO guidance framework for testing genetically modified mosquitoes, the taxonomic classification of mosquitoes, the current landscape of development for LMOs containing engineered gene drives for disease vector control, a list of engineered gene drive systems and a description, as well as a list of terms. And just to highlight here, as was mentioned by the previous speaker, that these additional voluntary guidance materials are aligned with Annex 3 of the protocol, as was requested in decision CP-1010. They build upon existing risk assessment guidance materials and synthesize them together, and it takes a problem formulation approach. Also, as requested in the decision, there needed to be a specific focus on mosquitoes, and mosquito-related content has been integrated into text boxes throughout the text. So related content on mosquitoes can be matched up to the main body of the text. And just to also highlight that the document has been formally edited and made available again in the 6 official languages of the United Nations. Regarding the needs and priorities for risk assessment as identified by parties, as mentioned, we had 2 submissions of information processes, and we had the following submitted topics. We have living modified aquatic organisms, including algae, crustaceans, and fish, submitted by 2 parties. Living modified organisms containing engineered gene drives, submitted by 1 party. Genome edited plants and animals, submitted by 1 party. And operationalizing protection goals into useful assessment and management endpoints, submitted by 1 party. Also to highlight that another party submitted saying that they didn't believe that there was a need for further guidance on specific topics of risk assessment. And then to also highlight the recommendation from the process on synthetic biology, the multidisciplinary AHTEG on synthetic biology, suggested the topic of self-limiting insect systems to be considered in line with the process in Annex 1 to Decision CP-913. The AHTEG on risk assessment recommended that no further topics should be considered or prioritized for the development of further guidance without prejudicing the discussions on LM fish that are forthcoming. So in terms of the documents that are available and to keep in mind and some of the resources that are available, just to highlight document CBD CP-MOP 11/9. These are the formally edited additional voluntary guidance materials, and also to highlight the SABSTA recommendation, so following the negotiations from the subsidiary body. So this is SABSTA recommendation 26/5 on risk assessment. And then just to also highlight the information documents, so SABSTA document 26/5 on risk assessment, the report of the AHTEG, and then also the web pages that have been created for living modified fish and engineered gene drives. And also just to highlight, since the discussions will be talking about fish, and there was a previous process on that the study on risk assessment for living modified fish is also available for parties to use. So next month in Cali, Colombia, the negotiations will finalize for this intersessional period. And in terms of some of the elements that are being considered, it includes the acknowledgement or endorsement or taking note of the additional voluntary guidance materials. This also includes a process for parties and organizations and relevant stakeholders to submit on their experiences using these materials. Since the topic of LM fish wasn't open for discussion at SABSTA to be in line with the previous decision from the CP-MOP, so there'll be a discussion on whether there will be a process to develop guidance on living modified fish or not. And then there's also elements about other topics of risk assessment. So this includes the submissions from the parties, the outcomes from the horizon scanning process under synthetic biology, and in line with that process in Annex 1 to Decision CP-913. And also just to highlight that there have been several decisions that urge for a coordinated approach between synthetic biology and risk assessment under the convention and the protocol. So with that, I would like to thank you for your attention and thank the organizers once again for the opportunity for the Secretariat to support parties. And with that, I'll hand it back to the moderator. Thank you so much.
Thank you very much, Austein, for your presentation. I believe these presentations will be shared. And for those who are going to the COP, I will encourage you to go over the last slide, especially on the considerations for the negotiations. and prepare adequately, bringing in some of your peculiar national experiences, and not just as part of it, of course, funding support would come in through the JEF and other mechanisms and capacity building. But what are the challenges that we are facing now? What are the best practices we've seen? What are the lessons that have been learned? And how can it help the global process in handling the issues that are coming up? Will be very useful. Thank you very much, Austein. And we will go to the last but not the least presentation by a lady that's this is the second time I'm meeting her. Don't know, the second time virtually. I met coincidence at the gates of the UN, and her name is Dr. Barbara Glover. She's going to make a presentation on risk assessment of gene-derived mosquitoes from an African perspective. For those who know Asia and Africa, mosquitoes are a big deal. So scientists normally want to look for all options to address challenges in mosquitoes. Barbara Glover is an advocate for the effective harnessing of science, technology, and innovation to enhance socioeconomic development in Africa. With interest and expertise in strengthening the African science policy innovation nexus, Barbara currently serves on the Secretariat of the High Panel on Emerging AU High Panel on Emerging Technologies at the African Union Development Agency, which is called AHTEG. In this role, she worked towards harnessing scientific advancement for the sustainable development of the continent. Her passion lies in promoting collaborative efforts that leverage technology for positive impact across diverse sectors. Her current work builds upon experiences working since 2016 in 8 African countries in identifying, harnessing, and implementing emerging technologies for Africa's socioeconomic development. In this role, she supports conducting research, analyzing data, and strengthening capacity on topical issues such as gene drives for malaria elimination to the integrated vector management program. In addition, she engages in evidence-based analysis of science, technology, and innovation papers, drafts policies and recommendations, and contributes to the creation of and implementation of capacity building program aimed at fostering a culture of innovation among the youth. So Dr. Barbara Glover also, I believe, will make presentation from a youth perspective, which is very important for the COP and the COP-MOPs coming. And I wish you well in your presentation. Over to you, Dr. Glover.
Thank you very much, Dr. Alex, for the kind introduction. I hope that you can all see my screen in presentation mode. So today I'm here to present on the African perspectives when it comes to risk assessments for gene drives. Thankfully, the previous speakers have given a good rendition of what risk assessment is, so I'll just focus on what we as Africa would be working on in that space. I wanted to introduce our organization just for the benefit of the new audience. So we are the Development Agency of the African Union, and we are tasked with 5 mandates. Our first mandate is to coordinate and execute regional and priority projects. We also are tasked with strengthening the capacity of all 55 African Union member states. We are also tasked with advancing knowledge-based advisory support to our African Union member states, undertaking resource mobilization, and also serving as the technical interface when it comes to engagement with our partners like yourself in our African countries. In terms of innovative vector control tools, which includes gene drives, we have 3 main targeted approaches within the African Union Development Agency. First of all, we seek to create awareness, and the essence of this awareness is that you can't regulate something that you do not understand. And so we want our member states to have a good understanding of the technology in order to make the right recommendations on regulations. We also then support enabling policy and regulatory environments, having an understanding that these tools are emerging. They need to be sort of domesticated within our national and regional frameworks. And then we further do that through effective capacity strengthening initiatives. So why gene drives? Why are we interested as Africa in gene drives? One major thing is the fact that there's an impact of disease like malaria on the continent. If you look at the 2022 malaria report, 94% of all the malaria cases reported were from Africa. And 95% of all the malaria deaths globally was from Africa. And the sad thing to note is that over 78% of all those deaths were from children under the ages of 5. And so this is really a concern for us as Africans, and we seek to find the best innovative tools to address this challenge. The African Union High-Level Panel on Emerging Technologies was established by the African Union presidents, heads of state and governments to give recommendations on innovations and emerging technologies across the continent. And one of the technologies that they recommended was the gene drive technology, having an understanding of this malaria burden. There have been several technical reports that have been released by the panel, and based on those reports, we were tasked as the development agency to implement programs that will ensure that this technology is effectively harnessed. We believe that it has a potential based on the thorough research and recommendations of the high-level panel to address malaria on the continent. And we also have an understanding that there is a cost to Africa when it comes to doing nothing about the technology or holding on to exploring innovative tools that can address malaria on the continent. And so we are looking at pushing forward dialogues, engaging with the relevant partners to ensure that technologies of this nature are adequately harnessed to address malaria across the continent. There are several opportunities for Africa when it comes to gene drive research. There have been certain countries that have led this research so far. We have countries like Burkina Faso and Uganda that are actively engaged in utilizing this technology to address malaria and other vector-borne diseases associated. We are also strengthening international partnerships beyond the continent, primarily around knowledge transfer. I noticed in the comment section and the Q&A some questions from the audience on Latin America, considering the fact that when it comes to LMOs, there have been active research in that space. And yes, we have strengthened our partnerships with certain countries like Brazil, Colombia, Indonesia, to learn from them on some of these initiatives, how they were effectively regulated, and how they can be transferred to gene drive research on the continent. We are also actively building capacity on the continent because we want solutions that are driven by African expertise, technical expertise. Our messaging as African Union Development Agency has always been that trust your scientists, African leaders trust your scientists. And so we ensure that capacity is built on ground to be able to address this. And we also work on strengthening biosafety frameworks where there are gaps that are identified to be able to support some of these innovative vector control tools on the horizon. Admittedly, there are challenges in the African context when it comes to harnessing technologies like gene drive. I earlier mentioned the regulatory capacity needs. Some African countries, when it comes to biosafety frameworks, are lacking, and we need to strengthen or even in some instances fully develop new biosafety frameworks for these countries to be able to effectively assess and regulate emerging technologies like gene drives. There's also ecological and biodiversity concerns with understanding that ecology on the continent isn't harmonious, and that predicting long-term environmental impact is a bit nuanced, and there is need for further research to be able to understand what those concerns might be. And then also the essence of public engagement. One of the lessons we've drawn from our engagements with countries outside the continent has been the need for intensified public engagement and the application of ethical considerations when it comes to the harnessing of these technologies. In order to do this, we have supported countries with the development of biosafety laws. We have programs like the African Union Biosafety Network of Expertise that have actively supported countries in the development of biosafety laws. We are also strengthening or building the capacity of our regulatory bodies, establishing of institutional biosafety committees, providing technical training, providing evidence-based scientific research outputs that would enable them to effectively regulate these technologies. We also have an understanding that this technology is mobile. We have mosquitoes that are released as a technology. And so there is a need for moving away from national-based approaches to regional approaches, harmonizing biosafety laws and regulations, at least at the regional level, to ensure that when a gene-drive technology is deployed in a certain country, the neighboring countries have applicable laws or favorable policy and regulatory environments to be able to support the deployment that is happening in a target country. And then we are continuing on with our technical capacity strengthening initiatives with our partners to be able to address any technical gaps around gene drives. Now, moving specifically into the risk assessment framework, well, there have been a lot of detailed presentations on it already, so I'm just going to skip through. But as I earlier mentioned, we have African biodiversity that is quite diverse, and so we need to look at further research on unintended ecological consequences in the various habitats, and there has been active engagement of African researchers on addressing any possible hazard identifications or any doubts that anyone would have on the release of these technologies based on possible hazards. We are also looking at exposure assessments. So when African communities are exposed to the deployment of these innovative vector control tools, what that would look like and whether there are any risks that should really be looked to be considered. And of course, setting into place risk management protocols that would help to build the capacity of our national regulatory bodies on gene drive. There is also the public perceptions that we need to expand on, ensuring that we deal with any limited understanding on the gene drive technology. For instance, 2 weeks ago, we had a landscape assessment validation report for Burkina Faso, where we're looking at having an understanding of the perspectives of the public when it comes to this technology. So we brought in civil society, we brought in the parliamentarians, we brought in key actors that are usually not in the room when it comes to discussions on gene drives, to have a good understanding of their public perceptions of these technologies. What are their fears? How do we allay them with emerging or existing scientific evidence and facts? We need to look at informed consent protocols when it comes to the deployment of the technology in certain areas. Transparency in our research, ensuring that our research outputs are out there for anyone that has any allays, concerns, or fears to be able to read on and be informed about. And most importantly, ensuring that African voices are adequately reflected in global discussions. As I earlier mentioned, when it comes to diseases like malaria, we bear the highest burden, and it's important that frameworks or policies that are being developed that will be of most benefit to us will have our voices most adequately reflected in those discussions. For Africa, we are developing a co-evolutionary approach when it comes to gene drive risk assessment. This is where you're ensuring that you are balancing innovation and safety in the realm of genetic technologies. I would, in the next slide, further expand on that. So the co-evolutionary approach is to ensure that regulatory frameworks evolve in tandem with technological advancements. We don't want to stifle innovation because of fears with limited regulatory capacity. We want to ensure that there is that co-evolutionary approach when it comes to regulation of technologies like gene drive. We want to foster innovation on the continent while ensuring that there is safety through adaptive governance. For instance, in the February African Union Heads of State Summit, there was a decision that indicated that within our integrated vector management approaches, we need to inculcate in the risk safety monitoring of the tools that are deployed. So in as much as we are encouraging innovation, we are also ensuring that all safety protocols are in check. Ultimately, this is to ensure balance. We want to prioritize our public health outcomes. We want to look at, do you want a child under 5 dying every 5 minutes from malaria, or do you want to look at certain concerns that may be addressed with science and innovation? And these are our deciding factors when it comes to the harnessing of technologies like gene drives. We also want to support continuous monitoring and adaptive regulations, having an understanding that these technologies are emerging. I think in a year or two to come, we'll be talking about other technologies other than gene drives, for instance. We want to ensure that our regulatory systems are adaptive. And in so doing, we build the capacity of our regulators and our health systems. We bring together other actors that are non-traditional when it comes to health product regulations. For instance, with gene drive, bringing together the biosafety experts, bringing together those from the ministry, environmental impact assessment experts, and together building that capacity to be able to harness some of these technologies. And of course, as earlier mentioned, our regional collaboration initiatives, bringing together the various countries for shared learning, for mutual learning, peer understanding of what has worked in countries within Africa and even outside Africa. In June, we took together some African experts to Indonesia, where there was an exchange between the ministries and even the ministers at the ministerial level to have an understanding of what worked in the deployment of technologies like dengue to address dengue in Indonesia, and how Africa can learn from these initiatives. Wrapping up, the key benefits of our co-evolutionary approach as Africans is to promote responsible innovation, mitigate any risk in real time because of our adaptive approaches to risk assessments, continue to engage our communities, make them a part of our decision-making when it comes to risk assessment, and ensuring that we align with international standards around risk assessment. So the last slide, I believe, moving into COP, we are going as a unified African voice because we have an understanding of the burden of malaria in Africa. We want to ensure that our risk assessment frameworks are streamlined across the continent, all 55 countries. And our anticipated outcomes will be that we'll have strengthened biosafety protocols that are tailored to some of our African needs. And also we would increase international collaboration, which is why I believe this webinar is a very important step, increasing engagement with partners outside the continent and sharing our perspectives in that regard. And of course, also furthering our capacity building focus as Africa. Thank you very much for listening in, and I hope that these perspectives have been useful to the dialogue. Thank you.
Thank you very much, Dr. Barbara Glover. You've raised issues on gene drives, issues on how we could take a continental approach to risk assessment and related governance matters. Colleagues, we have, per the 100 minutes given me, we have 5 minutes, but the Secretariat guides that we should have a Q&A for 20 minutes, so I crave your indulgence. A lot of questions have been posed on the Q&A platform and have been answered. I will need some guidance before we start this session from the Secretariat of who's organizing this meeting. Are we going to, do you want us to just continue with what is on the Q&A platform, or are you going to open for live questions? I stand to be guided before I continue. Since can I wait a minute more, or I should go ahead with the way forward?
Okay.
I'm not hearing from anybody. Hello, okay, go on.
Yeah, thank you, Dr. Alex. Well, from the number of the Q&As that are posted, well, first, I would like you to pay attention to those questions, Alex, and then you can choose one or two, and then after, I would like you to open questions on the floor, to the floor.
Okay, thank you.
Thank you.
I thought so also, but still looking at the time. Yes, so there's a question for Austein or the organizers to put up the last slide that Austein presented on preparation towards the COP, as it will guide some of the questions. So that would be done. At the end, I will pose a question or recommendation to Dr. Glover. You don't need to answer, but it's a long-standing question I've been asked for the past almost 20 years, back to the African Union, which could help in your preparation as parties from Africa. Yes, so as from the open site, most of the questions I've answered, there are 5 there. I will pose the first question and ask Brinda, Dr. Carino, and if others, if Dr. Vibha wants to add, you can add, because a very broad question. It says, what are the biosafety concerns related to gene drives? Is there any case study in this regard? What are the biosafety concerns related to gene drives? And are there any case studies in this regard? So Brenda and Dr. Carino, and then Vibha and the rest can also both add to.
Thank you, Dr. Alex. The biosafety concerns are the same that you would have for any other LMO. So you look at the sequences, you look at the organism itself, you look at the sequences in the construct that is inserted, or the traits, the source of those traits, the source of those sequences. So that's for the the proteins that are expressed in the insect that are not normally present in the insect. If there are any, or any deletions, you look at what the deletion would do. You look at the receiving environment to see what is the role of the organism in the wild type, what is the role of the organism in that space in the ecosystem, and what the modification might impact in terms of predators, prey, any other organisms that depend on it and that interact with it, decomposers, anything like that. It's the same that you would do for any other GMO, to be honest. So the biosafety concerns are pretty much the same. Allergenicity, toxicity, things like that. A case study, probably you can get some, there's not a direct case study, obviously, because I said there's no engineered gene drive or organism in the field right now. So the closest case study would be anything that is in containment. Otherwise, to some extent, it's not a GMO, but biocontrol if you want to look at spread and dispersal. And if you want to look at a GM mosquito, then there are some GM mosquito products available being commercialized in Brazil. So it would be nice if you could speak with the Brazilian regulators to see what they looked at. Again, that's not engineered gene drive, but it's a GM mosquito. Thank you.
Yeah. Thank you. Any other comment from Dr. Carino or any other the presenters? Okay, if we are not getting further comments, there's a question here, I don't know whether it's a question, but I'll post it. The person wants clarification on the different terms of LMO, gene-edited organism, and gene-drive organism, and the CBD context. What does it mean? LMO, gene-edited organism, and gene-drive organism. The person wants clarification. Any representatives can chip in on that.
Perhaps I can assist a bit. So regarding LMO, there is a definition in the Cartagena Protocol under the use of terms. Regarding engineered gene drives, the additional voluntary guidance materials provide a good description of engineered gene drives that was developed by the ad hoc technical expert group. And then regarding genome editing, there are diverging views on whether genome editing is if an organism produced through genome editing is considered to be a living modified organism or not. This relates to that definition in the Cartagena Protocol. Different parties have taken different views or perspectives on that and consider them differently. So unfortunately, I don't know if I could be more.
Austein, I think he's gone. Okay, maybe he's finished. Basically, these definitions, most of it are negotiated terms or scientific terms. What you do in your national regulatory system is your definitions of what you mean as part of the laws that you set up. I remember years ago in the Ghanaian context, the first draft of the law saying that in this context, LMO means GMO. And then they give the definition of what they mean. So in the CBD context, some of the terms, LMO was negotiated, gene-edited organism, there have been some semblances of definitions, and even gene-drive organism, there's some definitions. But you can crystallize all these and then come out with, unless there's a harmonized, like it was done for the LMO, where there's a clear-cut definition, the party guided by the science can also define in the context of the regulatory framework. I think I'll end it there. There's a comment for Dr. Glover that the problems we're elaborating are similar in Latin America, and the risk is of not doing anything and maintain a status quo for vector-borne diseases at COP16. I think this Maria's comment is something that the two regions can have further collaboration and discussions on how to take this forward. Before I open for the live questions, I also pose the comment I wanted to make to Dr. Glover, and also, it's a more of a recommendation, also the other regional groups that might be here. The Biosafety Protocol is set up both for state parties and regional parties. Currently, only one regional party is involved in the CBD and its protocol, that is the EU. But you will notice that it helps them sometimes in coming to harmonize decisions. I posed this question to the EU almost about 15 years ago. Because what happens is that for the parties, they have the obligation, as it is a no reservation protocol, to implement. Whatever the UN does, the AU, other regional group does, they are doing it as observers and facilitators. So I remember when I was focal point in Ghana at the ICCP3 before the protocol came into force, I made a comment which was per national discussions, and the African group was not happy. But I had been, so to speak, giving marching orders to give Ghana's position, not Africa's position. So there was a bit of confusion. However, if the Africa was a regional party, we could have taken that discussion in a different manner, because then it's a regional obligation also. So for the regional groups, what can be done to implement Article 14 of the Biosafety Protocol on bilateral and multilateral agreements, we could lead to regional parties taking part in the discourse, especially in the discourse, especially in areas of harmonized approaches. Don't answer is something that you need to think about. Thank you. I will now, with the permission of the Secretariat, open up for live questions, but we must close within the next 14 minutes. So the floor is open. Are they allowed? Okay, I see Joseph Okori, I believe from Nigeria, who has raised his hand. Joseph, are they able to unmute their mics? Okay, so you can unmute and ask your question.
Can you hear me?
Yes.
Good afternoon, everyone. I'm Joseph from Nigeria. The question I wanted asking have actually been answered, and I really want to appreciate the issue that has been raised, and we looked up as a country to be able to look for some of this issue in the forthcoming COP. Thank you.
Thank you. Thank you, Joseph. I see Lillian Chimpepo's hand up from Malawi. Lillian, over to you.
Thank you. Am I ahead? Yeah. Okay, so thank you very much. I just want to thank the presenters for a clear and in-depth presentation, which are very useful as we prepare for the COP and COP-MOP in Cali. I had posted my question, which was just asking clarity from the presentation by Brinda, where she mentioned about the self-limiting insects as example, some of the technologies which are out on the market. So this is also another topic which the MATEC recommended for an additional guidance document. And during the presentation, it was highlighted that self-limiting insects, self-sustaining insects, and the self-limiting was like one of the type of gene drive. So I just want clarity whether the self-limiting insect is also gene drive, is one of the gene drive organism. And if she, and if there can be some examples so that we can make an informed decision as we go, whether we really need an additional guidance document or whether the present voluntary guidance on risk assessment of gene drive living organism is sufficient to cater for the self-limiting insects. Thank you.
Okay. Brenda, over to you. Lillian is also a member of the AHTEG.
Yes.
So just for information.
Yes,
Brenda,
thank you.
over to
you. Thank you very much, Lillian. So what I showed in my slide was that there are applications of engineered gene drive organisms that could be self-limiting. That's just to say that some of them can have traits that can be engineered to be self-limiting. That does not mean that all engineered gene drive organisms are self-limiting or should be self-limiting, or that all self-limiting insects have a gene drive in them. Any combination is possible, Lillian. If they have a engineered gene drive in them, then the current document should be sufficient. In the case of purely GM or LM self-limiting insects, these are already some of their types are on the market, depending on how you define self-limiting. So the GM80s that is available direct to consumer in Brazil can be considered self-limiting, scientifically speaking, because its first iteration, it's a population suppression type of application where you release a mosquito and eventually it dies, and then subsequent generations are sterile, so the eggs don't hatch, right? So that could be considered self-limiting because the trait will eventually not be present after the mosquitoes that you release die, and no babies are born because the eggs don't hatch. So the trait doesn't persist. And then the second generation of that product, and the trait exists for a couple of generations, so 6 or 7 generations maximum, and then it it goes away because, like I said, it's a population suppression and there's a sterility factor and a high fitness cost. Similarly, if you look at the Burkina Faso sterile male release for mark release recapture, you will see the same thing. It's a sterile mosquito, so it's by definition self-limiting in that the trait cannot spread because the mosquitoes are sterile. I hope that answers your question, Lillian.
Lillian,
Yes, yes. Thank you very much. Yeah, thank you.
thank
It's
you very
really
much.
helpful. Yeah.
Okay. There's a lady called Maria Mercedes Roja. She's availing herself for anybody interested in learning more about the new ERA guide, I believe from the Latin America region. You are welcome to contact her at her email address, which she has provided in the chat. So over to those who want to follow up on that. If I've also asked for the projection of the last slide from the CBD. Any question on this before I round up this meeting or clarification? I am not getting any comment, so I believe it's very clear. Now let's continue with our preparation towards the COP. As I said in the last webinar on synthetic biology, another broader question we need to ponder is that Article 29(4)(e) allows for amendments, allow for further guidelines and further changes based on the current situation in the way the protocol should be implemented. Is Annex 3 now due for amendment, or we go by the approach of further guidelines on thematic issues that would come? Because the Annex 3 is the general principle of risk assessment, so it can hold. Are there any new issues that can make our decision-making at the party level more science-based? So there are several issues that guided by Article 29 for para 4E, you can look at. I've already talked about also measures for the regions to be able to have a better harmonized approach in case or if they were parties. Finally, for me, we are happy to see a continuous changes in the process from a single event risk assessment to multi and all kinds of interactions and capacity that is coming up. I had the privilege of joining this process during the negotiation in 1994, and I've seen a lot of changes. One nagging thought which I would, with no offense meant, is knowledge transfer. As faculty and experts age, sometimes knowledge is not transferred or people are not willing to learn, but jump the gun and therefore tend to not to present facts properly, even in negotiations. Negotiation skills are extremely important in presenting facts and experiences in international meetings, and it's something that those who are going to the COPs must hone their skills on and come up really with issues that would be of benefit to them. The JEF is also going to have the opportunity to make a side event on looking at the challenges you face in assessing JEF funding for the Biosafety Nagoya Protocol. I encourage parties to join that side event, I believe on the 24th October, and make your own cases on what is happening, whether a single window approach or status quo as it is. The final point also is that the JEF is agreeing to support your obligation under Article 33 on national reporting, and is providing resources, which is not from a national location. So the door is open for all parties that are eligible to request UNEP for endorsement formats and sign off. The JEF wants this project approved before the end of this year. So strive as much as possible to get this to UNEP before end of October, because when the JEF approves, we cannot add other countries to the funding process. So that is where I'll end and hand over to the organizers. We are very happy for your presence. We had at least more than 150 people throughout the presentations and the discussions. We wish you well as you prepare to go either a short journey or a long journey to Colombia. Thank you very much. Over to you, KIPABiC.
Thank you all. Yeah, thank you all. What an
Thank you.
insight.
And
What-
goodbye.
Yeah, what an insightful and engaging session this has been. First, I'd like to extend our deepest gratitude to Dr. Owusu-Biney for expertly moderating today's discussions. Your insight and leadership were crucial to the success of this session. And to our distinguished speakers, we offer our sincere thanks for sharing your invaluable expertise and experiences. Your contributions have greatly enriched our understanding and sparked meaningful discussions. We look forward to continuing and building on these conversations in the future. Our heartfelt thanks to all participants from around the world. Your active engagement, thoughtful questions, and contributions have significantly enhanced the quality of today's discussions. We hope the insights gained today will be valuable in the important work you do. Finally, I would like to acknowledge the efforts of the organizing team at KIPABiC, our technical advisor, Dr. Alex from UNEP, and Dr. Vibha Ahuja of BCIL India, and our project focal points in the 4 participating countries. It's your efforts and collaborative efforts made this event possible. As you exit the webinar, you will have the option to provide feedback. Please take a moment to share your thoughts on today's session. Let us know if there are future topics or areas you'd like us to explore. Your feedback is very invaluable in helping us shape future initiatives to better meet your needs. Thank you once again for your time, preparation, and continued support. Thank you.
Thank you.
Thank you.
Thank you.
Thank you so much. Have a good day, everyone.
Thank you all once again.
Have a good morning in northern part of the world.
Thank you, everyone.
Thank you.
Bye-bye. Thank you.